Well, next I'd like to welcome Dr. Sara Grove with the University of California, Santa Cruz. And Sara will be talking about the impacts of drought and Scotch Broom on Douglas-Fir seedling success. So take it away, Sara.
I'm here. Can you hear me?
Yes.
Can you see me?
Yes.
Okay, great. And can you see my talk in regular?
What
can you see for my talk?
Not yet in the presenter view. Now it is. Go ahead.
Okay, great. So all the work I'm presenting today was done in collaboration with Ingrid Parker and Aaron Aiello at UC Santa Cruz and Karen Habensack at Northern Arizona University. And in case you're not good at paying attention to webinar talks, no judgment here, I get it. I'm going to give you the punchline right off the bat on my title slide. And unfortunately, it is that Scotch Broom invasion increases the impacts that drought has on Douglas-Fir stress, survival, and growth. And for the rest of my talk, I'm just going to describe how we came to that conclusion. Okay, but before I talk about that, actually, I'm going to start my talk with a really big diversion and tell you a little bit about some work that Ingrid and Karen and I have done over the years, looking at Scotch Broom impacts that are mediated through soil legacy effects. So I began working on Scotch Broom a little over a decade ago now. And I worked as a first-year graduate student. I was working with Karen and Ingrid at Joint Base Lewis McChord on a project that was aimed at understanding how to manage Scotch Broom in a forestry context. And I think Ingrid showed this same picture. We did a project in 5 different Scotch Broom invaded clear cuts. They were really big clear cuts and they had really intensive Scotch Broom invasion. We removed the Scotch Broom from the 5 clear cuts and then we planted in thousands. I think it was upwards of 10,000 seedlings across the 5 clear cuts. After we removed the Scotch Broom, we planted in the 10,000 seedlings and came back after a growing season and found that nearly 90% of those seedlings had died. So this was super alarming and problematic for our study, but it was also really intriguing and interesting. And we thought that maybe Scotch Broom had changed the soil environment in some way that inhibited Douglas-Fir establishment. And that sort of interest or suspicion really catalyzed my dissertation research. And we followed up with a bunch of studies looking for these soil legacy effects. Before I show that figure, let me just describe one of those studies. So following this finding at JBLM, we did a study in 9 different clear cuts in the South Puget Sound region. These 9 clear cuts had, part of the clear cuts were really invaded by Scotch Broom, but part of them were occupied by native vegetation. And so what we did was, similarly, we removed the vegetation, we removed the native plants, we removed the Scotch Broom, and then we do what we do. We planted in our Doug-Fir. We came back after 1 and 2 growing seasons and measured our Doug-Fir survival or Doug-Fir mortality. And this time we were explicitly looking for legacy effects. And what we found is that Doug-Fir, there was nearly twice as much Doug-Fir mortality where cytisis had occurred, providing pretty compelling evidence that there are these strong legacy effects of cytisis that limit Doug-Fir establishment. And we went on to study what these legacy effects are. We studied things like how long does it take for these legacy effects to establish following invasion? How long do they last or persist after you remove Scotch Broom? And I thought I would share a little bit about what these soil legacy effects are. So as I think 2 or 3 people have already said, Scotch Broom is a legume. It adds nitrogen to the soil. This nitrogen can persist in the soil even after you remove Scotch Broom. That is an important legacy of Scotch Broom. With nitrogen fixation can be a phosphorus demanding process. So phosphorus can be decreased. This decrease in phosphorus availability can persist as a legacy also, that can persist for several years. We've also seen that Scotch Broom invasion can decrease the abundance of ectomycorrhizal fungi. These ectomycorrhizal fungi can be important to ectomycorrhizal plants like Douglas-Fir. So when you have a decrease in their abundance, that can decrease the performance and establishment of ectomycorrhizal plants. And importantly, these soil legacy effects can facilitate the secondary invasion by other exotic species. And there's a nice paper out by Carter et al. I think Tim Harrington is a co-author on that paper, where they've really nicely demonstrated how Scotch Broom can facilitate sweet vernal grass invasion and other exotic grasses. And we've spent so much time thinking about and working on these soil legacy effects is because we think that they contribute to positive feedbacks that ultimately reinforce Scotch Broom invasion and limit ecosystem recovery to pre-invasion conditions. And we just think that it's important to be aware of these soil legacy effects if you're a broom manager or someone who's doing restoration in broom invaded systems. And I'm happy to talk more about them anytime. But the Scotch Broom can also impact Douglas-Fir through just good old-fashioned competitive exclusion as well, where it just can better compete for limiting resources than the native species that it displaces. And I'm going to be talking for the rest of the day just about competitive exclusion under drought conditions. So in Western Washington, particularly in the Puget Sound region, we know that summers are expected to be hotter and drier. On the left here is a little image. Where's my cursor? There it is. That shows, sorry, can you see my cursor? That shows.
Yes.
Yes.
Okay, great. That shows temperature change on the y-axis and time on the x-axis. And what you can see in the red and green here is that with high or low carbon emissions, depending on how that turns out, there's going to be somewhere between 3 and 12 degree increase in temperatures over the next 75 years. And with that, there's going to be a 22 degree decrease in summer precipitation, and that's with either the high or low carbon emissions. And this is problematic for plants because water demands are highest for plants in the summer. And the summer is when plants are doing, need most of the water. That's when they're doing most of their growing, putting on their biomass, defending themselves from things like herbivores, reproducing. Plants are doing most of their work in the summer. And that is already when water is the most limiting resource for plants. We know that drought causes plants considerable stress, but we also know that plants have, remarkable adaptations or strategies to deal with drought stress. But what we need to be thinking about, and what we are thinking about, is whether or not invasive species are going to magnify drought stress to such an extent that it decreases the survival of plants and has population level impacts on native plants. And I have to say that at first it may seem like that of course invasive species will exacerbate drought stress, but actually it's pretty hard to predict. And that's because invasive species interactions are dynamic. And how invasive species and native species interact under drought conditions will depend upon the drought tolerance of both the native and invasive species, as well as the nutrient acquisition strategies of each of these species. For example, plants that are fast growing, plants that are fast growing and are really excellent at competing for resources during wet years, they might do really poorly during dry periods. And maybe Scotch Broom would be a species like this. Simultaneously, plants that are better, that are maybe slow growing, like woody species, that have better resource use efficiency, they might be good competitors during drought years, but poor competitors during wet years. So a species might be a good competitor during a wet year and a poor competitor during a dry year, and vice versa. So there's one complication in that invasive species are often thought to have high phenotypic plasticity, and that's one sort of been an explanation for why invasive species are invasive species because they can move from environment to environment and sort of do well in whatever environment you throw them in. And Jennifer Funk and Peter Petusek had a nice paper in 2007 that showed that a handful of invasive species that they tested were both very successful at being competitively dominant in both wet and dry, under both wet and dry scenarios. So it can be very hard to predict. All right, so what we do know about Doug-Fir is that even in the absence of Scotch Broom, Doug-Fir mortality can be really high during drought years throughout the Puget Sound region. We also know that in the South Puget Sound region, there are a lot of sites that are sort of they're sort of drought prone, I'll call it. These sites are what foresters call, they refer to them as low site quality, and they have they generally have a low fertility that is due to poor soil moisture holding capacity. And what we've noticed is that these same sites, with poor soil moisture holding capacity, they also have a lot of Scotch Broom in them. And we don't know if the Scotch Broom is there because Scotch Broom has good good tolerance for low soil moisture or for other reasons. But it certainly warranted further study, understanding how Doug-Fir and Scotch Broom interact in these low soil water environments. Okay, so as the first pass at understanding how drought and Scotch Broom are going to interact, we did a greenhouse experiment, and our primary question we asked was how do the impacts of Scotch Broom change with added drought stress? And we asked a couple of additional questions. We asked how do soil microbes increase or decrease stress under drought and broom invaded conditions? And I'll talk a little bit more about our hypotheses and provide some background to that question momentarily. We also asked, can Doug-Fir from drought-prone sites tolerate broom invasion and drought better than genetically improved seedlings? And I'll talk about that towards the end of my talk. Okay, so we used a factorial design for our greenhouse experiment where we grew Douglas-Fir seedlings with and without drought, with and without Scotch Broom, and with and without a live field soil inoculum. And then we measured Doug-Fir survival, we measured Doug-Fir physiological stress response, we measured the growth of surviving seedlings, and we measured mycorrhizal colonization of surviving seedlings as well. Okay, as I said, we were interested in how soil microbes interacted with drought and invasion. And it could be that soil microbes could alleviate stress caused by invasion and drought. That could happen if mycorrhizal fungi sort of came to the rescue. Mycorrhizal fungi can form a sheath around the roots of Doug-Fir and then extend hyphae out into the soil. And this hyphae extends far beyond where the roots themselves can go. These hyphae can access soil, water, and nutrients and provide those resources to the plants that the plants would otherwise not be able to obtain themselves. Mycorrhizal fungi can also provide protection from pathogens, and together these services can increase a plant's competitive ability. But soil microbes can also really exacerbate plant stress, and that's because soil also contains plant pathogens, and plant pathogens can reduce a plant's ability to acquire soil resources like water and nutrients. Soil pathogens can reduce plant growth rates, and they can reduce plants' competitive ability. So we don't really know if soil microbes are going to exacerbate or alleviate plant stress caused by invasion and drought. And we set out to test that. To do that, we collected field soils from 3 uninvaded Douglas-Fir forests. I sent my dream team out here, 2 toddlers. We collected soil from, oh yeah, I already said that, 3 sites. And then we brought these soils back to the greenhouse and we made mixtures that contained 20% of either live or sterilized field soil mixed with 40% potting soil or 40% sand. Okay, so what did we find? We found that Actually, first, let me start by orienting you to these rather complicated figures. So this is a 2-panel figure. On the top panel, we have plants that were grown in the live soil. And then on the bottom panel is plants that were grown in sterilized soil. On the y-axis are the proportion of plants that survived, and then we're going to start by looking at the plants that were grown in just the wet soil. Okay, so what we found is that in the absence of drought, broom really had no effect on seedling survival. So we look here, like basically 100% of plants, both in live and sterilized soil, survived to the end of the experiment. Drought, what we also found was that drought alone decreased the rate of seedling survival. So if you come over here, in both live and sterilized soil, you can see that the blue bars are much shorter with drought than the blue bars when the plants were watered, and same with invasion. With drought, there's basically very little survival when you have invasion and drought, which is our most important finding, right? Broom had strongly magnified the effect of drought, which was really an unfortunate finding. Okay, and then comparing the top panel to the bottom panel, we found that live soil inoculum further reduced Douglas-Fir seedling survival. Meaning that this is not, well, I'll leave it at that. Okay.
Your
And
5-minute
we
warning,
also
Sara.
measured how plants responded to how plant stress responded to drought and broom and microbes. And to do this, we used the fluorometer. The fluorometer measures the proportion of chlorophyll sites that could be photosynthesizing to the proportion that are actually photosynthesizing, and it's typically used to indicate plant stress. And these data were collected pre-dawn, and a team of undergrads and a grad student collected most of this data. And I'm super grateful for them for doing this data collection. It was really hard to be in the greenhouse by 4 a.m. And they collected this data 2 to 3 times per week. So let me walk you through these results. Okay, so invasion, first we're going to compare this dashed line to this line right here. And essentially what this is invasion only. So that, let me orient you to this grass a little bit. So we have FVFM ratio on the y-axis. The higher the number, the more, the better the plant, the less stress the plant is having. The lower the number, the more stress the plant is having. And on the x-axis is the number of days since we imposed the drought treatment. 0 is right about here. And then the brown color are the plants that experienced drought, and the wet color are the plants that received plenty of water. And I'm going to point to I'm going to use red arrows to indicate the interesting comparisons. And this is going to be our first interesting comparison. And what this comparison is invaded, comparing plants that were invaded to plants that were just droughted. And what we learned from this comparison is that invasion by itself was more stressful than drought was by itself. But this is probably just because broom was present for a good 6 months before we imposed the drought treatment. So likely that the plants just came stressed from broom before drought was even imposed. Okay.
This is your 2-minute warning, Dr. Grove. 2-minute warning.
Okay. Is that in the absence of broom or drought, soil microbes actually decrease stress, but that in the presence of broom and drought, soil microbes increase stress. Problematic. And then our main finding is that drought increased the impact of invasion on plant stress. When we looked at the effect, we also measured plant growth, and what we found here is that in live, we were only able to measure the effect of invasion on plant growth, because by the end of the experiment, most of the plants that received drought were dead. But what we found is that in live soil, Douglas-Fir were much larger in the absence of broom. So the difference between invaded and uninvaded here. In sterilized soil, however, the negative the negative effect of broom was really minimized. This suggests Scotch broom changes soil in ways that reduces Doug fir's competitive ability. And this can happen from a number of different ways, and we were not really able to test the mechanism. We're not sure which one of these processes were at play. It could have been one or all of them, but it could be that Scotch broom reduces the abundance of mycorrhizal fungi. I'll talk more about that in a second. It could be that it increases the abundance of soil pathogens, or it could be just that soil pathogens have a bigger impact on the seedlings because they're so stressed from the drought and the invasion. We did look at mycorrhizal fungi, and consistent with our previous findings, we did find that invaded soils, Scotch broom really reduced the abundance of ectomycorrhizal root colonization on the Douglas-Fir roots. And this mutualism disruption could have been caused directly by the suppression of fungi from the Scotch broom, or it could have been caused because the plants were so stressed from the Scotch broom that the Doug fir were unable to provide their symbionts adequate carbon. And so the fungi were lost through indirect effects.
I hate to cut you off, Sarah, but we're going to transition to the next talk.
Okay, that's fine.
Maybe 30 seconds.
Okay, I'm just going to quickly tell you my conclusions. There was importantly, we learned that competition for water is an important mechanism that limits Doug fir success. Broom by itself did not reduce Doug fir survival, but did increase stress and reduce growth. Drought by itself had negative impacts on Doug fir survival, physiological stress, and growth. The impact of drought was increased by broom invasion. Soil pathogens and competition with broom may have additive effects. And at first look, we learned that the wild type Doug fir from drought-prone sites may not perform better than the genetically improved seedlings that are typically used in Doug fir forestry. Much more work is needed, and that's why we're taking this study into the field in collaboration with Green Diamond Resources and Forest Genetics Consulting. And many thanks to the people who did that 4 a.m. work.